Thanks for bringing juice.
Sorry?
Thanks for bringing juice.
Oh, yeah, of course.
Hey, awesome.
Um,
Yep.
so, in this semester, sorry, I haven't put back to office hours.
Okay.
Plug everything in. Why do I feel like there's so much less stuff?
I think it's we don't have the
The little pointer thing.
Okay, so I'm okay with that. I'm gonna forget our conversation. So it's probably best to send me an email and then drop by so we can figure out like what that would represent and what that would mean.
Yeah, sure.
Alright,
I just want to plug it in.
thank you so much.
Okay, it's on.
Professor G,
Yes.
I was ask if you were still looking for students to write with your
Yes. What was your topic?
I'm doing microbiota on public and learning.
Okay.
So yeah, like, can I shoot you an email and then
Sure.
see if you need anyone else?
You need to download it, right?
Yeah.
Download it? That's fine.
Yep. Just send me an email and
Okay, sounds good.
no problem.
Should we set up OBS again just so it'll screen it? Like if we go straight into it, because it'll go onto the other one. You're right. Yep. It's fine. You can just pop up afterwards.
Were you supposed to just stay up here?
Sorry, Duffy. Do you mind if I ask you a question?
Sure. Go ahead. Yep.
So, on the assignments section,
I'm going to be talking about the assignment and when it's due. So if you're asking if it's due on the 7th or the 6th, I'm going to talk about it.
okay, also, can I ask you another question?
You just did. Okay, go.
Okay, my notes on it for some reason is not working at all. Can I just take notes and email it to you?
I guess that's okay. You don't need to email it to me though.
You don't have any big screen.
Okay. And is this the way that we were doing it?
This is a standard way
Okay.
of the pull everywhere on either side.
So
and the
what
slide.
were we doing in the last lecture? Do you remember?
Last lecture we had the slide on all three since there was no pull everywhere.
Oh, you guys are using that, sorry?
Slide on all three, no plug
Yeah.
everywhere. So can we just have the lecture slides going across the
Yep,
top and then the
absolutely.
pull everywhere on the two smaller ones?
On the two smaller ones?
Yeah.
Perhaps.
Try it.
You good with just this mic, or do you want
Yep, no, that's it for me.
Rockstar it out the last
Nope,
day?
sorry. Nope. So is there a different configuration that we can get here?
There you go.
Nothing.
No.
Oh wow.
That doesn't
Is it because of the
Yeah,
black line
that
and the
doesn't
maple
make
center one?
No, we're just wondering if.
we can't change these bottom ones without flipping the top one, right?
Okay.
Yeah, I think we can.
So can we just get both?
We could go 2 and 1.
Yeah. Let's try two.
No, I think that
you know
only
what-
splits a screen.
Do you know what he wants to do?
Yeah, yeah, yeah.
Okay, cool.
It only splits a screen. It doesn't like it doesn't
No, I was
actually
wondering.
like.
Yeah, no, I was wondering if it would split. Oh no, so just still make it smaller, right?
Yeah. Oh, they're opposite. That makes sense. Right. Okay, that makes sense. That makes sense.
I'm going to remember nothing.
That looked a bit strange. But I guess
Interesting.
Okay.
Interesting.
Okay, I'm going to go see.
Are we good for streaming?
sorry. Yeah. Should be
Okay, sorry, and I'm gonna ask you to just stick around because once they switch
okay.
out,
Yeah, we need to switch out the file. Yeah, we got it.
Would you prefer if I do my announcement now or after the break?
That is up to you. Sorry, active mics. Sorry, how do I volume control wireless mic?
I feel like a barista. Wait, what? I feel like a barista mixing my two things.
Sorry, when we gotta get going. So
Yeah, you want to start now?
yeah, I think so. We actually have a lot of stuff to cover, so we should just.
I'm ready.
Okay.
I'll go through my slides.
All right, folks. Hi.
Thank you, by the way, so much, Dr. Drew.
I'll do my announcement after the break.
It's up to you. So you can do it like really quickly now if
Yeah,
you want.
that's
Okay, folks. Hi. Wow. Nicely done, Akiza. So we have a lot of announcements before we actually get going with the lecture today, and it's we're trying to keep it fairly short. We have an announcement from CASA. Is that how you pronounce it?
Yep.
And then we have our course ambassadors. And there's a presentation by the course ambassadors. Okay, you need to hold that.
all right. Hi everyone, my name is Isha. I'm from the Cognitive Science and Artificial Intelligence Student Association. So quickly, cognitive science is the study of the mind, and we take insights from all different disciplines like psychology and neuroscience and computer science to explain the mind. So we have a really exciting conference coming up in February about vision and visual perception and how that affects cognition. We've invited speakers from Canada and the United States from their different fields such as linguistics, computer science, neuroscience, and psychology to come and talk about vision and visual perception. Information about the conference is online at cogsci.ca, and on Facebook you can Google Vision 2020. Tickets are on sale now, and if you have any questions you can come and ask me. All right, thank you.
Hi everybody, so it's our last class. It's my last class this semester, which is super exciting. So we just quickly wanted to talk about how you can study for the final exam. So up here, I've written down kind of the steps that you could take. I feel like many of us, when we're studying for an exam, we just kind of panic, then cram, and without having really much of a strategy. So here, you actually want to start with sort of a diagnosis, whether that is like a practice test that you have, or you want to self-test by going through the lectures. And you want to chunk up all the lectures that you have into bigger themes and topics. And you want to start with the ones that you're feeling the least comfortable with. You don't want to go through all of them 1 to 12, one at a time. You want to start with the ones that you're least comfortable with, and then spend the least amount of time on those you're the most comfortable with, which sounds really obvious, but I feel like most of us don't do that. And then you want to spend most of your time consolidating the information, which will be in chunks of 1 to 2 hours. And I'm sure you've learned of this in your other courses as well, but you want to use as many active learning techniques as possible, whether this is helping, like trying to teach yourself, teach your friends, make diagrams, make charts, create questions as well. And then if you have consolidated the information well enough, then the memorization shouldn't take much longer, especially in a course like HMB300, when it's so heavily concept-based. All of the memorization will come from making those charts and consolidating the information. And then finally, you want to end with a self-test. And then this is kind of overboard. I can't imagine myself studying this much in a day, but what I wanted to, the main point I wanted to make with this is that you want to study your big themes and topics in larger chunks of time from 1 to 2 hours, and then you want to keep memorization to quick chunks throughout the day.
So when we were talking, whoops. That was a good question. Hi everybody, can I just get your attention? The mic is not really it's not working really well. So really quick, some advice our learning strategists had was to take care of yourself. It was very, study plan can really help you avoid having an unbalanced study, focus a lot on 2 lectures and then cram the rest. But if you're not following through, can you guys hear me at the back? Yeah? All right, perfect. If things are not working for you, your plan is not working, that means there's something either wrong with your plan or it could possibly be you. You could be a little bit burnt out, you could be a little bit tired. So it's always important to be reflective and see if you're missing anything that you need to do. Take a break, get some food. Because I know when I get studying, I kind of forget to take a break and eat. Always double the time you think you need to get something done. It's easy to underestimate than overestimate. Most importantly, take care of yourself during this time. We really want to prevent burnout, so plan some pockets of joy. So that's like whatever makes you happy, even if something small as getting Starbucks. And it's also important to always carve some time to plan every day because you're likely not to finish everything that you've planned for the day. So you're probably going to shift some things, and that is completely normal. And do mindful activities. Let's say you wanted to go get a coffee at a nice coffee shop. Do it with the idea of savoring this moment and enjoying it. Don't be in the moment is what I'm trying to say. And then I have some few gifts for ideas to how to spend your break, have a snack break, dance break, change subject, and change it up with a study location.
Friends, no. We're done in 30 seconds. This part's probably the most important part. So guys, so as much as we work super hard in this course and we think we're dying, Dr. Ju has put in so much effort and time into making this course wellness focused and helping us get as much of this course as possible. So we have a little gift from all of us to him. Thank you so much, Dr. Ju.
Woo! Thank you.
Thanks so much. Okay, we're good? All right, thanks so much. We'll get going. All right, so thanks so much for that, and thanks for a great semester so far. We still have a little bit of material to cover, and I'm also going to point out a few things to you as we go along, so please make sure that you're able to follow along. So, there were a couple of questions that I had been asked because I kind of skipped over them. I said I'd come back, and this is our last lecture, and so I need to make sure that we're all sort of on the same page related to what you need to know for the final exam. And again, I will point out a few things to you in the very last slide. So you only have 15 multiple choice. Some of you are asking, I'm not really good at multiple choice test questions, do you have a strategy? Well, there are only 15 multiple choice test questions, and there are 10 short answer questions, and then there are 3 longer, one-page essays, and I'll go through all of that at the end. So just keeping in mind, so it'll be okay. Most of the material is stuff that we will make sure that you're in good shape for. So one of the things that I've been asked the most about is, I had mentioned Krebs binding protein, or CBP, versus phosphorylated Krebs or phosphoserine on Krebs, and it does form a dimer. It does bind to the cyclic AMP response element, which is this Cre sequence, and that is found directly on the DNA, the target DNA. And this Krebs is something that gets phosphorylated, and when it sits on here, it becomes a transcriptional factor. If you look over on this diagram here on the right, it's showing you exactly the same thing. So we have a Krebs dimer. It's phosphorylated on serine 133. But in addition to that, we also can have a Krebs binding protein, or CBP, that helps to further activate the transcriptional regulation through this Krebs. So this is really what I want you to focus on, the Krebs dimerization that leads to the transcriptional activation of various downstream genes. And what we've been looking at in particular is how if we get this activation of Krebs, we will usually see things like survival, proliferation, differentiation, and primarily through the upregulation of BDNF, which has been the molecule that we've been the most interested in. So hopefully that will answer the questions around the serine 133 phosphorylated Krebs, and it does form a dimer, and it does bind and activate the transcription of various genes, and you do not need to worry about the ones that are shown here. The ones that I had on the earlier diagrams are the ones to focus on. So I also wanted to go back and share this particular slide with you, and this is really to get across the fact that if we were to look at one of those factors that gets activated after BDNF binds, one of those factors was known as RAS. And RAS will activate MAP kinase, it will then cause transcriptional regulation through Krebs, etc. And if we create a construct or a transgenic animal and we overexpress RAS, so think about what the implication of that means. So we are activating the BDNF pathway, And we're specifically activating the BDNF pathway that's involved in growth. So there's survival, there's growth, there's proliferation, there's differentiation, but RAS and MAP kinase is specifically involved in growth. And one of the things that you can see is if we were to create a transgenic animal and we are using a synapsin 1 promoter, which means that this genetic construct that we are creating here, this is the genetic construct that follows behind it, and this is the RAS that we're talking about. If we are expressing this construct, it will only be expressed in neurons. So only neurons have this. And this makes it really, really interesting because that's what we've been studying throughout this course. And one of the things that you'll see if you look, this is the wild-type brain, and this is an age-matched animal. And one of the things that you'll see if you look very, very carefully, even though they're age-matched, by simply overexpressing RAS within neurons using the synapsin 1 promoter, so that, again, BDNF activates a number of different pathways. One of those is involved in growth. It's not on this slide, but it was on a previous slide. And what you can see is by making neurons grow bigger, the brain itself grows bigger. BDNF will have that capacity. And what we're looking at here in blue is instead of using GFP, which we would use nowadays, the authors of this paper that was published in the late 1990s decided to use this LAC-Z gene. And this stains sort of a blue color when you give it the appropriate substrate. And if you look, every single part of the brain is in fact bigger than the wild type animal. So overall length, if you look from the olfactory bulb all the way back to the occipital lobe, the overall length of the brain is bigger. The width of the brain is bigger. The hippocampi, which is what is being indicated by this little arrowhead, it's bigger. In fact, the cerebral cortex is bigger. Everything gets bigger. So this BDNF pathway, by stimulating it and just using the downstream genes that are involved in growth and differentiation, we can actually see neurons getting bigger. So BDNF has these roles, and this is one of the roles that is shown on this particular slide. So do you have any questions about this or anything from the previous slide before we go on? Or anything that I can go back and re-explain for you? No? Seeing no questions, I will go on and talk about this. So these slides, the next few slides, I'm hoping that you'll connect with what we started talking about at the end of the last lecture. I'm going to tell you in advance that you are going to have questions related to placebo. I can tell you in advance that you will have multiple choice test questions related to single nucleotide polymorphisms and placebo responders. I will tell you in advance that on the short answer questions, you should be thinking about these slides as well. So this has good representation on the final exam. And one of the things that we were looking at is we know that one of the best places to look at placebo is the pain response. And so when we look at taking away pain, we call that analgesia. And this feeling of not having pain is placebo-driven. And we talked about how in Alzheimer's disease patients, that as part of their frontal lobe starts to deteriorate, and because the frontal lobe is the first part of the descending opioid pathway, so do you remember that pathway, dorsolateral prefrontal cortex? And then it's going on all the way down to the level of the spinal cord. At each of those different synapses, we are releasing opioids. And so if we block this pathway, or we lose part of this pathway, we lose the placebo effect, which uses the exact same pathway. So if we go back and examine this model, where we have a painful stimulus, we have a reward on one side of the bars, the animals have to press their face up against the bars to get that reward, but it's painful. So they have this conflict where they want to get the reward, but at the same time it's painful, and we can measure how long are they up against the bars. And one of the other things that we know that we can do is we can also give them an analgesic that directly activates and stimulates the opioid pathway. It causes opioid release, and as we do so, they feel less and less pain. So we give them an analgesic, they feel less pain, so they're going to spend more time up against the bars. And we can do this twice. So we can give them an injection of the control substance, which we call phosphate buffered saline or PBS, or we can give them the true analgesic like morphine, and then we can try to fool the animals. We can get into their belief system. So the first time they got an injection, they didn't feel so much pain. If the next time we give them PBS instead of morphine, will we be able to see that even though they're not getting the analgesic, that they're still spending as much time up against the bars because they think they should feel less pain? Do you remember that experiment from last week? So the other key thing that hopefully you remember from last week's experiments was the fact that we can block this entire placebo effect by giving different drugs like naloxone, so it's an anti-opioid substance, or we can give naltrexone. And so if the animal's belief system involves the opioids being released more, we should be able to block it by giving an injection of naloxone. And in fact, this is what they did. The results are actually very, very convincing. So what you're looking at here is the further along these axes that we are, the longer the time the animal is spending up against the bar. So for PBS, and they're given PBS twice, they feel the pain, and so they're not spending very much time up against the bars, and we're looking at the actual test of giving them PBS versus the very first time they're given PBS. And again, the axes are not important. What you're looking at is if they're spending more time, we should see more dots going this way. And in fact, they're all clustered at very, very short time intervals. PBS doesn't do anything for them. They feel the pain. However, if we start off by giving them morphine and we activate the analgesic pathway, we activate that descending analgesic pathway, and they believe that the second injection, which is just PBS, so there's no morphine, we're not directly activating, they're activating it themselves through the dorsolateral prefrontal cortex, we actually see that they are spending more time up against the bars on that second trial, which is why we see this spread of data along this axis. This result actually disappears, so we don't see this increase along this line. It goes back to these very small clusters that are closer to the bottom if we give it naloxone. So naloxone reverses the effect. So naloxone, if we were to give them or have animals that have been treated with morphine and then given PBS and then are treated with naloxone, that blocks their placebo effect. And this is what we're seeing here, that naloxone actually reverses those trends. So it's not very high on this axis as it is over here. So do you have any questions about this? And again, one of the things that you're going to probably ask, it's not a really tight cluster. And it's not all the rats that are responding. It's about 30 to 40% of the rats that are actually showing the placebo effect to begin with. That's not any different than humans. So we know in humans that there is a genetic effect, there's a psychological effect, and so this is why it's not a perfect cluster down at the origin like you have at the top. So again, before we go on, and because you know that you're going to be asked questions on this, do you have any questions before we go on? Yes? So Miriam, it depends. So there's some context. So at the top, yes, we're looking at 2 injections of PBS. The second one, we're also looking at morphine and then PBS. And then the bottom one is slightly different. Yes, that's correct. Any other questions that I can answer for you? And again, you will have to remember like the overall effects of different drugs like naloxone and others on your final exam. You will also have to remember all of those different single nucleotide polymorphisms. There are like 2 pages worth of them, so please make sure that you spend time going over them. There is more than one question related to the single nucleotide polymorphisms and the placebo effect. How many of you at this time of year are stressed out? So I see that most of you are like really tired. Are you stressed? Yeah, stressed out. How many of you are starting to feel sick? A little bit, some of you. Do you think there is a correlation between feeling stressed and feeling sick or getting sick? Does it affect your immune system? And how does it affect your immune system? Inflammation? Could, potentially. So this is sort of an emerging field. It's kind of a really cool field if you think about it. It's only been around since about 1998, so it's been around about as long as we've been studying the placebo effect. And this is sort of the field of psychoneuroimmunology. So there is an impact of a belief system, so feeling stress, your brain, how it handles stress. Something that is stressful for you might not be stressful for me or for your neighbor, but we know it affects your brain, and it also affects your immune system, and it affects a variety of different systems. And we kind of have a better idea of how some of this might work. I'm going to tell you in advance that you don't have to go back and memorize this pathway because you probably have already memorized it in other courses. But this is sort of the key thing that we are looking at, this release of cortisol, the stress response. When we want to measure the stress response in a quantitative way, we look for levels of cortisol. The higher they are, the more stressed out you are. And that's psychological stress, it's physiological stress. And we want to understand like how all of these different stress factors can trigger this pathway, and how by doing so, by working on different areas of your brain and your belief system, how that can directly impact your immune system as well. And there are some key factors, and they're all kind of interrelated. This next slide is one that I want you to make sure that you spend a little bit of time on. So you'll have a one-line answer from this slide, you'll have a multiple choice test question from this slide, and you might need to incorporate different elements of this slide in your longer answers where you have a page to be able to answer all the material that you know. So we're looking at two different effects. And I'm going to start over here on the left. So we know that stress, psychological stress, so thinking about all the essays that you have that are due by Friday or Saturday morning, thinking about the exams that start either Saturday morning or on Monday, thinking about how much you're in debt, thinking about all these other factors. This is stressful, and it directly impacts part of your prefrontal cortex. And as a result, you will initiate this stress response. And the stress response, because it is going to cause the release of cortisol, and here on this particular slide, it's indicated as corticosterone. They're slightly different, but I'm going to use them interchangeably. I'm going to say that cortisol is equivalent to corticosterone. It's going to have a number of effects, both directly on the brain, as well as on the peripheral immune system. And there's some really fascinating things that start to happen. And one of the things that we know that happens is that if you are under continued stress, you are actually going to reduce the production of BDNF in your brain. And you can think about all of the implications of what that means. And we're not talking about short-term stress. So short-term stress is like 10 minutes at a time when you're like trying to catch that bus to make sure that you're getting to the exam in time. That's sort of short-term stress. Once you catch that bus, your cortisol levels go back down. But if I were to look at your cortisol levels now versus the beginning of the semester, I'm guessing that your cortisol levels have stayed pretty high for about at least a week, maybe 2 weeks. For some of you, maybe like a whole month. And that's not good. And that's sort of where you are going to start impacting the production of BDNF within your brain. So why is that particularly bad? Well, one of the reasons why we think that this is particularly bad is that it does directly, through the release of cortisol, it dysregulates some of the immune functions within your body. So one of the different types of immune cells is this natural killer or NK cell. And this NK cell stops functioning as an immune surveillance cell, so you're more likely to not be able to clear off different pathogens. And in addition to that, it also decreases the production of interferon gamma. So your immune system, because of these elevated and prolonged levels of elevated cortisol, will start to have a reduction in the activity of natural killer cells, as well as a reduced production of interferon gamma from those cells. And again, if you're thinking, well, once I get into my postgraduate program, everything is going to be wonderful, one of the things that you should make note of is that particularly in med school students are typically the population where blood gets drawn a lot, and they're the students that a lot of these studies are based on. And this is a prospective study that actually showed that for med students undergoing stress, that they actually saw all of these effects happening. And these stressed med students are the ones that have this suppressed immune response. They also get very sick during very stressful periods of time as well. The reverse. So this same prospective study showed that if you find ways to relax, similar to what Bakiza and Rena had suggested to you earlier, it's not sort of just pie in the sky. It's not just about, well, it might just be a good idea. There's a lot of studies that have gone in to show that taking time out to reduce stress, these stress busters, if you're wondering, like, why are there so many of them on campus at this time of year? This reduction in stress is actually incredibly important. So it's to make sure that you're not getting sick physically as well as mentally. And so these same prospective studies, so they followed them for 2 years. One of the things that they showed is that relaxation and de-stressing also enhanced natural killer cell activity, meaning that you're able to clear off any pathogens that might have entered your body. Now, if you look at the potential mechanisms, and I'm going to draw your attention over here, there are two different things to keep in mind. Some of the different white blood cells, which are indicated here for you, and you'll see that they're a different size, and if you are able to de-stress and you're able to relax, one of the things that you'll see is that they will enhance the production of LFA. This leukocyte factor antigen 1 is incredibly important for allowing cells to enter into different compartments and different tissues, and they will upregulate the production. Your immune cells will upregulate the production by being able to have like relaxation, reducing cortisol levels. All of these things will enhance LFA within the white blood cells, and these LFA producing cells, the leukocyte factor antigen 1 producing cells, are able to enter into different tissues, including the brain. So your brain is also a tissue that can upregulate the production of a different factor, which is known as ICAM1. So ICAM1, if you're looking on the left, near the frontal cortex, one of the things that happens is that ICAM1 levels within your brain increase. So here's a question for you that you might want to hang on to, something that you have to take notes down for. If you're thinking about ICAM1 and you know anything from any of your other courses, where would you expect to see ICAM1 levels being increased? So your brain is not just neurons. What other types of cells might you find within your brain? Microglia could be other cell types. What else goes through your brain? Blood. So what makes the blood go through your brain? Blood vessels. And so ICAM1 is believed to be increased within the endothelial cells that make up the parts of the blood-brain barrier, allowing immune cells to also enter into your brain. And again, there are lots of reasons potentially for that, especially as you start to lose sleep. There might be reasons for having more white cells white blood cells entering into your brain. This will ultimately lead to an enhancement of BDNF levels, so it does benefit your neurons. It's not really clear if there is a link between immune cells entering your brain, activated immune cells entering your brain, productions of things like interferon gamma and BDNF. They're not all linked together. We don't know what steps actually cause these, or if they're just two things that happen because there's a reduction in cortisol levels. But I do want you to make sure that you're spending a little bit of time on this slide. You will have to know what ICAM1 is. You will have to know what NK cells are. You'll have to know what LFA does. And again, a little bit of a tip for you. It's very similar to what you had on your term tests. So it's not just tell me what factors might be increased on white blood cells. It's more likely that I'm asking you if we see an increase in LFA1, what might happen to the cells within the brain. So they might enter more easily, or we'll see greater numbers within the brain. Okay, so one of the things that we really want you to be thinking about, especially after today, I'm not going to see many of you around as frequently as I'd like to. I can't really make sure that you're not being stressed out, is that you are going to have a lot of stress, but you have to be able to manage that stress. And it's incredibly difficult and challenging, especially in a high performance sort of university like U of T. You always feel like you need to be on top of things. So hopefully you've been trying to manage your stress. Hopefully you have good sort of stress reducing techniques. And I wanted to spend the last few minutes going through some important slides that you need to be aware of, and also hopefully to help you out a little bit in terms of managing your stress. How many of you have ever taken a meditation or mindfulness class to practice it, not to learn about it, but to actually do it? Mindfulness, meditation, a couple of you. How many of you think that it actually works? There's a biological basis, like just like we look at, yeah, stress affects your immune system, I just gave you some proof. Do you think there's a biological mechanism behind meditation? Yep, okay. I think most of you are kind of saying yes-ish. All right, so the next few slides are ones that I want you to be aware of, and I'll point out what you need to know. If you're thinking a little bit about meditation, it's a little bit counterintuitive. And to be honest, when the first series of papers started coming out about 8 years ago, it didn't make a lot of sense to me at the time. Didn't make a lot of sense to a lot of individuals who are looking in neuroscience, because again, as we started off this course, we have traditionally been very, very neuron-centered. It's called neuroscience, but in fact, that's probably not the best way to look at it. And it turns out that meditation, although it has many, many beneficial effects, the things that it has the most effect on is probably oligodendrocytes and oligodendrocyte progenitor cells. White matter tracts are the ones that benefit the most from things like meditation. So if we were to look at one particular form of meditation, and this is known as integrated mind-body forms of meditation, and it's a type of mindfulness meditation. It's been shown by some individuals to actually reduce the levels of anxiety, and some of you might be encountering it, especially as you go to these de-stressing sessions. It doesn't actually take a whole month. It turns out that you only need about a week of this to actually see some of the benefits. And this type of meditation actually reduces that stress hormone that we just talked about, which is known as cortisol. The other thing that makes this really important is there's an area of the brain that is really important for you to be able to pay attention, and this is known as the anterior cingulate cortex. For this area of the brain is one that I'm going to spend a little bit of time on over the next half hour or so, and this anterior cingulate cortex actually increases in activity at basal levels when you have learned to meditate and when you practice mindfulness meditation. So if I went in today and I had no training at all, and I'm just coming in off the street, I am not able to focus on my studies, my mind kind of wanders, and it does, so I have like a mild learning deficit where it's really hard for me to concentrate on things. And so if I looked at it today, my anterior cingulate wouldn't be so great in terms of activity, but after about a month or maybe 2 weeks of mindfulness meditation training, my anterior cingulate activity at rest, basal levels, will actually be enhanced. And so we know that that's important. And it's especially important for students who are in school. The other thing, as highlighted in red, we also see that the white matter pathways that are leading into and out of the anterior cingulate cortex are actually much denser. There's more white matter there leading into and out of the anterior cingulate cortex as we measure in diffusion tensor imaging. So this is a really interesting finding. So how does that work? So how does just thinking about different things, or paying attention to the moment, or paying attention to your breathing, or paying attention to a very small aspect of your physiology, how does that, over the course of 2 weeks, cause physical changes that we can measure within your brain? Again, for me, when I first started hearing about this, I was very skeptical, like, no, that's not possible. Like, I think it's all kind of just wild science. It's like not very quantitative, but it turns out that it is incredibly quantitative, and it is something that I wanted you to be aware of. If you have the opportunity, one of the individuals who's an incredible leader in the field is known as Michael Posner. And Michael Posner is one of the first individuals to try to establish a set of guidelines and rules for how we can measure quantitatively, objectively, in an evidence-based way that we can all kind of replicate how meditation works to alter different properties of the brain. And so he's a leader in the field. He's in semi-retirement right now, but he is a huge person in the field of meditation. And so one of the things that he wanted to do, and this is a proof of concept paper that came out back in 2012, 2013, is that can you actually cause meditation to reduce smoking behavior? So again, we'll get into that in a moment. So I want you to think a little bit about what's stressing you out right now. Stress leads a lot to anxiety, and anxiety is something that we all kind of face, especially as we go into the final exam period. And these anxiety-related disorders, whether it's social anxiety, generalized anxiety, it could lead to panic attacks, panic disorder. All of these things start by having too much stress related to anxiety. So I want you to think about this in terms of relations to what you might be undergoing in the next week or so. So smoking is not a good behavior, as you would suspect. It affects a lot of different organ systems, including the brain. And one thing that sociologists and potentially some neurobiologists, some neuropsychologists as well, smoking and nicotine in particular as a part of smoking is actually considered a gateway drug. So it in and of itself is not harmful directly. It is physiologically bad for you. It will cause cancer. It will cause problems with your lungs. But its real danger, at least for sociologists, is that it lowers the barriers for other types of more addictive substances, especially in people that have access to these types of gateway drugs. And that's one of the reasons why neurobiologists are interested in this. So how does stress-induced smoking actually, how can we understand it, and how can we reduce all of this stress-related behavior? And one of the things that we know is if you're stressed out, you'll engage in different types of stress-related behaviors, right? So for me, if I'm like really stressed out, even though I know that I shouldn't be doing it, what kind of behavior do you think I engage in? I don't smoke, by the way. Quarter pounder, yeah, still can't get over the quarter pounders. Still do it. But it's not necessarily stressed. I think that's pure addiction. Anything else? Netflix. Yeah, I guess. Netflix, sure. Netflix. So I tend to procrastinate a lot. I don't know if any of you procrastinate, so I know I have to do something. I know I have a grant that's due. I know I have to write out a report, but I tend to procrastinate. And I know that that's wrong, but it's a type of behavior that I engage in. And it's really due to this lack of self-control. And I really want to try to fix that because I know it's wrong. I know it's bad for me in the long run, but I just simply can't stop procrastinating. It's still to this day, like procrastination is still an issue for me. And this idea of self-control. So imagine not being able to watch Netflix, but to study that extra 5-6 hours instead of just like binging on Netflix. Like how much better would you do in school, right? Instead of binging on Netflix, why not work on your review paper that, by the way, is due on Saturday, not due on Friday. So it's like due Saturday at 12 noon, not Friday at 12 noon. So how can we actually develop this self-control? And some of these studies, some studies by psychologists as well as some, so you seem very pleased that it's due like one day later. And that doesn't give you permission to watch a little bit more Netflix. It gives you permission to kind of spend a little bit more time on editing, right? So hopefully that will be something that you do. And there is some suggestion to suggest that your inability to focus, to concentrate, and the self-control might actually come as a result of a hypofunctioning anterior cingulate cortex or the dorsolateral prefrontal cortex, two areas that we've looked at before, and anterior cingulate we've spent time introducing just a few minutes ago. So if we can induce self-control and it gets rid of very bad types of behaviors that could lead to more consequences, and it could maybe be used in reducing stress-mediated behavior. So you probably know individuals. So I have a colleague who is teaching in physiology, kind of looks a little bit like me. When he's stressed out, he's like out in front of MSB kind of smoking. And that's his stress-related behavior, right? And can you actually use mindfulness to reduce stress-related behaviors, or for me, like get rid of the procrastination? So could we use mindfulness to kind of help in this aspect? The one thing that Bruce Posner had found out is that most people, like myself, and I'll have to be honest, when the first group of neuroscience students that I had who were coming in from like various disciplines, when I first started teaching, they were telling me, oh, there's this new thing called like meditation, and we think that it really impacts the brain. And I just poo-pooed it because I looked at the different types of studies that they were referring to, and there were lots and lots of issues with those types of studies, including like what Bruce Posner found. There's a lot of inconsistency in the literature, so there's no controls. So in science, we always need controls. I had pointed out to you earlier that, in fact, without that control that we had with the valine methionine 66-BDNF, we wouldn't actually have those results that we have here today. Controls are important. Many of the early studies on meditation had no controls whatsoever. It was all self-reported. There was no randomization. There were lack of biological endpoints. That means that I ask you, like, how are you feeling today? Similar to these questions, but I'm not taking your blood pressure. I'm not measuring your cortisol levels. I'm not measuring something biological. And so when you look at that as a scientist, you go, well, I don't know what kind of study this is. And this is the same thing that Bruce Posner had as well. So he wanted to kind of reverse all of that trend. He wanted to figure out ways to actually use biological endpoints. It's not self-reported. It's where you can actually measure something, quantitate something, have evidence-based data. He wanted to have randomization. He needed to have controls. And these were all really important in the types of studies that he wanted to revolutionize in terms of understanding the effects on the brain. So again, this integrated body meditation is like really something that had gained traction by the early 2010s, and he wanted to know if after a few sessions of this, could we determine, using biological endpoints and everything that we knew in neuroscience back in 2012, 2013, could we actually develop a rational evidence-based model for why meditation might or might not benefit the brain? So again, college-aged volunteers, it's not an outlier because college-aged individuals are still smokers to this day. In fact, nowadays, the bigger health issue is actually around e-cigarettes and vaping, and that's a huge issue in and of itself. But college-aged volunteers were used in this study that Bruce Posner had published. 27 cigarette smokers, so they're on average like heavy smokers. I guess they're heavy smokers, 10 cigarettes per day, and 33 non-smokers. They were randomized into two different groups that were looking at smoking levels before with relaxation training that's separate from integrated body meditation techniques, and they were given this training for 30 minutes every night for 10 consecutive sessions, and they wanted to know if you randomize these different individuals and then you go in and measure a biological endpoint, can we actually see a change after integrative body meditation techniques? And so one of the things that he wanted to do was to develop an objective measure. So normally, before 2013, a lot of individuals would just go in, they would do the training, and they were asked, like, how many cigarettes do you smoke a day? Well, I smoke 10 cigarettes a day. You do the 10 days of training, then you come back, and after that 10 days, you're asked, after like 5 cigarettes per day now. So that's the way that those types of experiments were done earlier. But Bruce Posner actually wanted to change that. He wanted a biological endpoint. So he took blood samples, and he actually measured the nicotine levels within these individuals directly after this integrated body meditation technique. And what you're seeing in red, you are seeing the levels at the very top before training, and these are all these different individuals. And then after 10 days of this training, the levels of the same individuals are indicated by this decrease. And this is sort of a relaxation technique. It's a similar type of technique, but it is not integrated body meditation. And then when you look at these individuals, this is their control, what you see is a lack of this big drop. So you can see huge drops over here, but you don't really see the same drops in the other control groups. If you were going to critique this data, what might you critique about this data? Is there anything that you might critique about this? Yes? The control results here on this slide are actually on the right. So it's a similar type of technique. They have smokers that were randomly assigned to a different type of relaxation technique. So they listen to music instead of like doing meditation, and the results are there on the right. And you see that before and after, there's almost no change. But looking at that data, do you have a problem with this data? Yes? Right, so they're supposed to be randomly assigned to these groups, right? So where are all the really heavy smokers on the right? Well, if this is before and after, so the red bars are before, why weren't more of these really heavy smokers that had lots of nicotine in their blood, why aren't there more of them in the relaxation technique on the right? So great observation. So Bruce Posner actually has no explanation. Scientists have asked him, and he said, well, it was just randomly, sort of randomly controlled, and so these were the groups that were assigned, and this is what we saw. But you can definitely see there is a big reduction in the overall levels after 10 days. Let me just go through this one slide, and then we'll take a break. If I can get to the next slide. Hopefully. Oops, nope. All right, well, the slide's not letting me advance, so why don't we go ahead and just take our break now. So, oh, never mind, it worked, okay. Don't go yet, just stay for like one more minute. So, I'm going to mention, because some of you are thinking, yeah, you know, I don't need to know this slide. Sorry, you will have a multiple choice test question on this slide, just in advance. I'm not trying to like make it easy for you, but at the same time, I know that you have a lot to study, and I don't want you to go later like, hey, where did that thing come from? Well, this is the slide that thing came from. Study this slide. So how did he look at the brain activity? So remember, we're looking at two different areas. Dorsolateral prefrontal cortex has been implicated, and so has the anterior cingulate cortex. One is involved in decision-making, dorsolateral prefrontal cortex. One is involved in self-control, the anterior cingulate cortex. And so one of the things that he uses is what is known as the fractional amplitude of low-frequency fluctuation. At rest, if you're stuck in an fMRI and you're not really doing very much, and your different resting networks come into play, so groups of neurons as you're just sitting there, not asked to do anything where you're actively thinking, you'll be able to measure these low-frequency oscillations in different brain regions, which is known as FALFF, or FALF. And this is normally used to look at the different brain regions that might be involved in things like post-traumatic stress disorder, if you have reduced attention, if you have mild cognitive impairment. Some people believe that after a concussion, you can go in, if you had like a resting state before, or you think that you're getting better from concussions, that you'll be able to see these changes on FALFF, and even potentially to see at resting state levels using this form of fMRI to see the early onset of things like Alzheimer's disease that we've talked about before. So they did this 2 weeks before this training had occurred. So remember, they're on it for like 10 days, and they saw a reduction in the overall smoking levels. And what they saw before they started the training, so don't get mixed up by this slide. So before they started the training, the heaviest smokers actually had very, very low activity in the anterior cingulate cortex. That means that they had very little self-control. So this is before they started, 2 weeks before they start. They had very, very little activity in the anterior cingulate and very little activity in the dorsolateral prefrontal cortex. So that means that for Posner, he is interpreting this as if you are a smoker, you have very little self-control. That second bullet point, first bullet point, you guys will need to know that. Are you clear on that? Yep, we're good? So 2 weeks after training, these areas, the anterior cingulate cortex, was dramatically increased in terms of the activity, but we don't see that increase, nor do we see an improvement in smoking after different relaxation techniques. Meditation helps, but not relaxation. And I'll just leave this one up here. So they're looking at the anterior cingulate cortex right here. And generally speaking, after 2 weeks of integrative body-mind techniques related to meditation, we're seeing an improvement in the activity in the anterior cingulate cortex, suggesting that for Bruce Posner, he interprets this as meaning that these smokers who have stopped smoking, that even if they encounter stress, they have more self-control with this meditation, and so he sees that as an increased activity in the cingulate cortex as opposed to 2 weeks prior. So they've improved their anterior cingulate, and therefore they've improved their self-control. So again, just to make it clear, you need to know what was on the previous slide. You need to understand how you can measure these changes. You'll see it on the final exam. I don't want there to be any surprises. Yes, it's from the last lecture. Make sure that you're spending time on it. We're going to take a 7-minute break if that's okay with you. Please come back after about 7 minutes. There's still a lot to get through, and I want to make sure that I leave some time at the end as well.
technically the end of the semester. It's in the syllabus. So I'm just thinking of the syllabus.
Thank you.
How are you? I'm okay. I'm just grabbing some snacks. You know, having a good time. Oh, he's here. He's Geo 6.
All right, folks. All right. So just to wrap up that first study, this mindfulness meditation, at least in Bruce Posner's hands, and by the way, it's not just Bruce Posner, so that study has actually been replicated. As far as we can tell, there are no major deficits to his study, although there's that caveat that I'm going to get to in the second bullet point. When we look at this, it seems to actually be working, and it's particularly well suited for different types of stress-related behaviors, whether it's like addiction to alcohol, smoking, taking opiates, etc. There's still a lot of caveats that are associated with this. The randomization process, at least in Bruce Posner's hands, as was pointed out by your colleague earlier, and the controls, they weren't really all that convincing. He did the randomization, but the controls, they didn't all start off with like really high levels of smoking, and so that lack of reduction wasn't something that is easily accounted for, at least in that model. The other thing that wasn't done in his study, because some of you might be asking, if you take a look, so like what was happening to this individual? So this huge drop and this huge decrease in the level of smoking, did that mean that their anterior cingulate cortex, was there a correlation with this individual where before it was like almost non-existent and then it became really, really strong in this individual? That correlation wasn't done, nor were there any correlations with this individual, so just a small decrease. It's still a decrease, but did their anterior cingulate not really change all that much? So it was more of an averaging out of the different data. So this correlation is not something that was done in his study. Other studies since then have actually gone and verified that the greater the level of strengthening in the anterior cingulate cortex, the better the level of self-control. All of that can be followed back to the levels of mindfulness training. So again, really, really interesting. The literature is still kind of mixed. Some people don't think that self-control comes from the anterior cingulate cortex. And if you've taken other psychology courses, some of you might have heard of the posterior cingulate cortex, the PCC, and some individuals actually implicate that area being more important for self-control. So Again, there are still some controversies in that area, but it doesn't mean that we should be giving up on this. And in fact, just a few years later, so he started that study 2012, published in 2013. Then this other paper that he started in 2016 and was published in 2017 actually looked at this in animal models, right? So can you actually do meditation training in mice? So if you can get mice to believe in a placebo, can you get mice to actually exhibit meditative behaviors? And this was sort of his next really big study, also in PNAS. And before you get to that stage, there are a couple of things that I need to point out to you in advance. So what's the one thing that you have to believe about the mouse brain and the human brain in order for his study to actually make sense? Is there anything that you have to take into account? Our brains are the same. So that's like the big thing. Like what we find in the mouse brain has to be directly transferable to the human brain. Our brains have to be fundamentally the same as the mouse brain. Okay, so that's like a given. But the key question, and why I have these figures down here, and apologies, I did not put that on your slide because I learned later that not all of you have had like a neuroanatomy course in your second year. Have you heard of von Economo neurons? So von Economo neurons are neurons that we only find in humans and great apes. We do not find von Economo neurons in the mouse brain. We don't see them. Everywhere we look, in the cortex, the cerebellum, anywhere, we don't see von Economo neurons. So is our brain fundamentally different than the mouse brain? Yes, our neurons are different. They're not the same. And these von Economo neurons, or the VENs that you see here, these are large. They're spindle-shaped, so the cell body kind of looks like it's elongated. It's a spindle. So think back to your physiology lectures where you learned about that spindle apparatus that sits in the middle of your muscle, sort of this elongated cell body. It's fusiform shape. We have them. Some large aquatic mammals like whales have von Economo neurons. Some great apes have von Economo neurons. Monkeys do not. Mice do not. Rats do not. And so maybe not all brains are the same. We will assume for the time being, though, just like Bruce Posner did, that in fact our brains and the mouse brains are linearly related. So they're just different in size. But it does turn out that we have unique neurons, like V-shaped neurons. This is a spindle-shaped neuron, only in human brains, not in mouse brains. A couple of more of these spindle-shaped neurons, we only see them in our brains, not in mouse brains. And again, we have some unique-looking, very different types of brain cells that mice do not. But let's assume for the time being that everything that we see in mice is the same in our brains. We can also test for anxiety in mice. So most mice, we can track them in this open field test, and many of you will go on to do fourth year courses and seminar courses and talk about and do critical analysis on different types of anxiety-related behaviors. So we can track these mice. It's done all automatically. You just set up the camera, you let the mouse go, and depending on the level of anxiety, the mouse will either stay close to the side walls, or they will explore a little bit more in the open field. We can also look at how active they are. And generally speaking, one of the things that we see is that if they are spending more time in the perimeter, close to these to the walls of the cage, they're more nervous. If they're less anxious and they're not as nervous, they will actually spend more time exploring the center of the apparatus. So this is one of the things that we use. And thigmotaxis is the word that we use to explain that behavior. When you are right beside a surface or a wall, you are exhibiting sort of thigmotaxic behavior. You need to have something that you are not worried is going to come out and attack you. So this thigmotaxis, or being next to a wall, is something that shows that the mouse is anxious. Okay, so we can measure anxiety in mice. There are other ways to measure anxiety as well. Some of you may have heard of the elevated plus maze, and this elevated plus maze is another way that we can go in and measure anxiety. So we have this two different arms. There are arms that you have encased in glass, And these closed arms are the ones that the mice will spend more time in if they're anxious or if they're feeling stressed out. And if they are feeling more relaxed, they will spend more time in the open arms, and they will actually explore more in the open arms if they are feeling less anxious. And this is, again, going back to rodents liking small enclosed spaces where they can feel a lot more secure. If they're nervous, anxious, they will spend more time in the closed arms proportionally than they will in the open arms. So we can measure that directly. One other thing, this is sort of an older technique, still in use. So I used it when I was doing my PhD because it was sort of the standard method. There's like one problem with that. So if you're stressed, yes, you are going to feel more anxiety, and yes, you are going to stimulate that amygdala, but what other area of your brain do you think is going to be affected, or what other behavior is affected when you are anxious? Is there any other behavior that you feel gets affected if you're anxious? Decision-making skills, right? And so here you have sort of this really interesting sort of intersection where the mouse has to decide, am I going to go straight ahead, to the right, to the left? And that decision-making involves the prefrontal cortex, and some individuals actually don't want to examine the effects of decision-making on it. So the newer types of anxiety-related controls are what are known as the zero maze. It's sort of shaped in a circle. There are no hard edges where you have to decide to go to the right or to the left. You still have enclosed spaces, and then you have open spaces, and then you just decide, am I going to go into the open space or stay in the enclosed space? So there's no decision about making a left turn, right turn. And so the zero maze is sort of the more modern way in which anxiety is tested. If you're in these closed arms, then you are going to be more anxious. And if you're in the open arms, you are feeling less anxious. And this, again, goes back to this. The way that this is, the mouse will have to make a decision that involves the prefrontal cortex. And if you're under stress, we all know that this happens. Like when you are stressed out during an exam, it's really hard for you to decide between choice A and choice B. They look really similar. What do you do? All that stuff. So this sort of takes away some of that. There's another way to measure anxiety as well. And this way to measure anxiety, so you put a mouse in the center, in that center chamber that you see here. And then we have these two small cages that you see to the right and to the left. They're about the size of one of the cups that you have up here at the front. And under one of these cages, we have a novel object, a thing like a Lego, and under the other one, again, they can see right through it, is sort of an unfamiliar mouse. Mice, when they're relaxed, when they're not stressed out, when they're not anxious, tend to be more social. And so you can actually measure, do they spend more time with the inanimate object, or do they spend more time with the mouse, as you're seeing on the right? And do you think this works perfectly just the way that I described it? No, just like the Morris water maze, it's not ideal because it sounds like it's going to make a choice, but sometimes it just stays right in the middle and it messes up all your data. So it doesn't go either place, and you can't really try to push it in one direction or the other. And think a little bit about like when you're feeling like really anxious, what do you kind of do? You kind of, instead of talking to someone, you'll kind of spend time with your cell phone, right? It's the same sort of idea here that when you're socially anxious, you're probably not going to spend time trying to get to know people, but you're actually going to try to spend more time with an inanimate object, your cell phone, and you'll find ways to go ahead and text. So one of the things that I saw a lot of my fourth-year students do was that they criticized, hey, this light-dark box is actually not a way to measure anxiety. It sure is. It's a great way to measure anxiety, and it's not just about place preference or conditioned place preference. We can actually look at light-dark exploration, and just as the name would suggest, if you put them right in the middle and they're feeling like really, really anxious, they're going to spend more time within the dark region. And if they're feeling like less anxious and less nervous, less stressed, they will actually do more exploration in a proportion or percentage-wise in the light region that you see over here as well. Conditioned fear training, like that sort of startle reflex. If you are anxious, this fear conditioning, you're much more likely to see more freezing behavior in an anxious mouse than you are in a mouse that's less anxious. So the amount of freezing is also something that you can use as a way to test for anxiety. And again, you might want to include some of these in any of your short answers. You do not have to worry about the details of them on your multiple choice, but for short answers, you might want to include some of these tests for tests of anxiety or stress in a mouse model. So again, I just went through a whole bunch of these things really quickly. Do you have any questions on this before we go on? Any questions, things that I can clarify for you now, things that weren't so clear? Yes? Yeah, we still do human testing. So like we looked at taking volunteers in these different types of experiments. And yes, it's still done. In fact, if you're in the psychology department, you probably get tested on all the time, right? Any other questions? Yes? Sorry? The criticism is that it's actually so there are a couple of things. So not all mice species that you're examining will interact and react the same way. It's really hard to get the light level to a consistent level that it's not going to scare all the animals, but if it's too dark, then they'll always go in there. If it's too bright, they won't go in there at all, regardless of how they're feeling. And so just getting the right levels is really hard. It's hard to also normalize that when you're reporting this, like you talk about the lumens that you're using, but even there, it's hard to replicate those findings. So one of the reasons why people don't use this more often as an anxiety test is that it's challenging to have replications of your experiment with someone trying to do exactly the same thing for anxiety in like a different country. All right. Okay, we're almost there, getting there. All right, so going back, we've already talked a little bit about this, and we've talked about meditation, and meditation may actually be as impressive as people say, and we also want to use this idea that meditation is incredibly powerful in an animal model, and these animal models are not so easy. And I'm guessing that most of you would never have been able to tell me ahead of time about how you would test for placebo in like an animal model system. I'm hoping that placebo response makes some sense to you now and why that works as a placebo, how you get that false belief system that the mouse thinks it's getting another injection of morphine when all it's getting is a control substance, and then all of these effects can disappear with naloxone. And if we see that in mice, we should be able to understand how that works in the human brain, and we have a lot of correlations there. And so Bruce Posner wanted to take this sort of to the next level. So in 2013, he had described that, yeah, we can actually find ways to measure in humans these physiological endpoints and these biological control measures. We can see things on an fMRI, and we know that meditation will cause changes to activity in different places within the brain. He still didn't really know back in 2013 how all of these changes actually occurred. He didn't know like what meditation was actually doing to the brain. All he knew was that I can see it, I can describe it, I have a biological endpoint that is not based on something that my subjects are telling me. It's not sort of just hearsay. I can measure this independent of what they're telling me. So he wanted to take this to the next step. And one of the things that he had hypothesized was that, again, this is before DTI became sort of the sort of favored child of like what neuroscientists look at now. So this is back in 2013. A lot of research had been done in the intervening 2 or 3 years, especially around meditation. And many studies were actually showing, unlike the initial studies, so when I first started looking at meditation in a neurobiological context, everybody was looking at the thickening of the cortex, increased gray matter, and these were sort of the early studies. Didn't make a lot of sense. What does that actually represent? Does it mean that the dendrites are getting bigger? Is there more neurogenesis? How's it getting up to the cortex? There are a lot of questions. When people started to re-examine this in the mid-2010s, they were actually seeing that there were more white matter changes, and that kind of made more sense to more neurobiologists. So you're not changing the number of neurons. The neurons aren't getting bigger, because that would be really weird, to be quite honest. But you could change the level of myelination to connect areas of the brain better than they were before. And so one of the things that had been reported in humans using different measures like fMRI or using EEGs was that if you underwent meditation, that you would be able to see an increase in the oscillations of firings of the neurons within the frontal cortex, which is known as theta rhythms or theta activity. And nowadays, one of the reasons why we're so interested in things like epilepsy, where we get uncontrolled bursting of the brain, or large regions of the brain bursting together at very, very high frequencies, is that we know that different types of wave activity in different areas of the brain actually change the brain in ways that we didn't think was possible. So if you're trying to consolidate a memory, you probably need these gamma oscillations or gamma ripples. If you want to induce changes related to meditation, you need theta activity. Theta activity is a feature of like meditation and deep concentration. And so this idea that if you look at individuals that train for like 2 weeks, we're not talking about like lifelong meditators, we're just talking after 2 weeks, you will see an increased theta rhythm in most individuals that have been trained properly just after 2 weeks. And this means that in the frontal cortex, when you are entering into a meditative state, you will actually see your neurons synchronizing together and having a wave pattern of about 4 to 8 hertz or 4 to 8 cycles per second. And that is a very characteristic feature of meditation. And so knowing this and trying to connect how we get increased myelination and maybe theta activity, and how this might relate to things like glucose metabolism, how it might relate to changes within the anterior cingulate cortex that he had looked at before, he wanted to know, okay, let's not worry about humans, because people are already doing that. Can I do the same thing in mice? The problem is, how do you get mice to meditate? Suggestions? Ideas? Meditating mice. Tell them to take Paul Whistle's course. They'll all meditate. Possibly. Anything else? So it's not easy. So his hypothesis and his working hypothesis is a little different, but I hope that you'll understand the logic behind this. I can't get mice to meditate. Sorry, it doesn't happen yet. Maybe 10 years from now, 20 years from now, we might have humanized mice. These humanized mice might be able to do it. Who knows? But right now, what we can do is artificially create theta rhythms in this area of the brain. And his simple sort of logic is, if I induce this activity within the area of the brain that we think happens in humans, can I see these white matter changes after inducing theta activity that normally mice would not enter into by themselves? If I induce it artificially in these mice, can I see less anxiety, number one? Number two, can I see increased myelination? And those were his two simple questions. Do you understand the logic of his experiment? Do you think it's a good experiment? Okay, you're reviewer number two, obviously. I think it's a pretty interesting experiment. So if we take a look at how this might be done, how is it possible? How could this be done? It's very, very challenging. So we couldn't have done this prior to 2009. The technologies didn't exist. It would be impossible. I would have to literally take off the entire front of the skull of the mouse, which is possible, and you can keep them alive. But then I'd have to put in so many different electrodes and have them firing sequentially And these electrodes are ones that mice would want to pull out if they have it. So it would have been really, really difficult. So I'm not going to be able to cover all of this this year in this course, but one of the things that they decided to do was to use a technique which involves gene transfer of a channel rhodopsin. You can use different flashes of light at 4 to 8 hertz and use optogenetics to actually stimulate a large area of the brain. I don't need to inject in a lot of different electrodes. And he was able to, using optogenetics in this area of the brain, get the entire frontal cortex to fire at 4 to 8 hertz. And after he used optogenetics to get this area of the brain to fire at 4 to 8 hertz, he examined their anxiety levels using the same types of behavioral tests that I indicated for you earlier. They were all decreased. Their anxiety levels went down if they had 4 to 8 hertz. In fact, if you tried different stimulations, so you tried 1 to 3 hertz, didn't work. They were anxious. If you tried higher frequencies above 8 hertz, so 9 to 15 hertz, they also did not show any changes in anxiety. Also, they saw increased myelination, probably due to the recruitment of oligodendrocyte progenitor cells. Their novel object location and recognition assays as a function of anxiety levels were also improved. Their cognitive levels, their ability to think, remember, was also improved dramatically as well. So all of these measures showed that by inducing artificially this theta rhythm activity in a mouse brain, so we can't get them to meditate, but we can introduce brainwave activity that looks like they're meditating, that all of that reduces anxiety, enhances cognition, and it makes the mice perform better. It was a very, very interesting finding. To date, in 2019, so I looked this up before coming to class, there's been no study to actually indicate this as being correct, and it's still a very powerful study to this date. This is the last slide that I need you to spend time on. So there is like one short answer question where it's like a one-line answer, and there's also a multiple choice test question based on this one slide. And I don't want you to think that, oh, it's the last slide, I don't need to worry about it. I need you to worry about it, and I need you to understand what I'm asking you to know from this slide here. I'm not going to be able to go through all of the optogenetic methods that he used. If you're interested and you take CJH, we'll go through some of that again in that course and how that method was developed. But there are two different ways that I can actually induce theta wave activity. And this is why I'm putting up this slide, and there are things that you need to know from this slide. The frontal cortex is huge. It's not like the hippocampus. The hippocampus, relatively speaking, is a small, very, very self-contained structure. The frontal cortex is massive. So it covers millions of different neurons, and there's no way to make sure that you're stimulating all of them equally. So one of the things that Bruce Posner decided to do was, hey, I can actually stimulate neurons two different ways. I can directly excite them, or I can block their inhibition. So normally there's a little bit of a break on these neurons. They have like inhibitory cells stopping them from firing. And if I remove that inhibition at specific frequencies, then I can get the brain to fire at like 4 to 8 hertz, that theta wave activity. And here is the cortex. So the cortex has 6 different layers, top to bottom. Top of the cortex, that's right under the skull, is layer number 1. Then we have layer 2, 3. We have layer 4, layer 5, and layer 6. And then underneath that, we would have the white matter, all of the axons coming down into the brain. This is sort of the normal topology. And if we look, and you do need to know this, and I'm not trying to be disingenuous, so I want you to make sure that you get a good mark on your final exam. And I also want you to learn this because it's important as a neurobiologist, that if we look at the inhibitory cells, and these are common in mice as well as in humans. So humans, we have one extra cell type, which is a von Economo neuron. It is an excitatory neuron. We have them, mice do not. But we have exactly the same types of neurons in our cortex. We also have 6 layers in our cortex. And so the mouse brain kind of looked like ours over here. So if you take a look at layer 2-3, we have all of these very unique looking cells. We have these bipolar cells. These are inhibitory. And the CB stands for the protein that is found in these inhibitory neurons, and this is known as calbindin. So whenever you see calbindin, it is an inhibitory neuron. So that means that this neuron, by definition, will release GABA. It will stop nearby neurons that it's connected to from firing. And does this look like it's going to connect to a lot of different cells? No, not very many, right? Just the layer above it, the layers below it. So layer 2-3, just very few. Then we also have sort of at the level of layer 3, almost at layer 4, we have this calbindin positive, therefore inhibitory, multipolar inhibitory neuron. And do you think this is going to connect to a lot of different neurons? It connects to more, but only on the same layer, so only like in layer 4. So it's a good and very interesting sort of inhibitory neuron, but it's also like very localized. If you look at this other calbindin type of inhibitory neuron, this is a beautiful neuron. And the shape of the neuron is what we describe, and then it can actually contain either calbindin or it can contain calretinin, which is another marker protein, also found in layers 2-3. And you can see that they have this double bouquet shape. So there's a bouquet of flowers going up, and there's this other bouquet-like structure that's going down. So do you think that this will inhibit a lot of cells? It does a better job than the multipolar, but it's still very restricted to the layers above it, the layers below it. Same thing for the calretinin positive one and the calretinin bipolar cell. But then we get into these really interesting cells. So they don't contain calretinin, they don't contain calbindin, and these are unique to these cell types, calretinin and calbindin, and these are found in these layers. As we go deeper, as we get into layer 5 or layer 4-5, we get this different type of inhibitory neuron, and instead of expressing calbindin or instead of expressing calretinin, we actually see these inhibitory neurons, therefore they express GABA, they release GABA, they inhibit their nearby neighbors, and one of the things that we see is that they express a protein known as parvalbumin. Okay, and these are found in layers 4, layers 5. Do you all see that? If you look at their connectivity, these chandelier cells, because they literally are the most beautiful cells that you will ever be able to see in the nervous system. If you ever have the chance to see them, like, make sure that you have a chance to see them. They're spectacular. It's like seeing Niagara Falls. It's just amazing, and they make all sorts of synaptic connections at multiple levels, the layers above it, the layers below it, but they also spread out. And then if we look at the basket cells, these are the ones that send huge, long projections to multiple layers. And so his idea was to actually use these parvalbumin-containing neurons to inhibit a larger number of connected cells, so he could control large numbers of cells, because by using the parvalbumin-containing basket and the chandelier, many inhibitory connections with so many nearby neurons, he could control them more precisely than he could with double bouquet cells or with the bipolar type of cells. Does that logic make sense to you? So those are the ones that he used. They're parvalbumin. I'm not going to lie, you need to know this slide, and you need to know all the different markers. And on top of that, do you think you need to know the different layers that they're found in? For sure. And I'm mentioning that because it's one of those questions that you can kind of bank ahead of time. It's a multiple choice test question. I'm not giving you the question in advance, but if you study this slide and you know the layers, you know the types of markers that you'd find in them, it'll be a question that you should find fairly easy. If you don't study it, it's not going to be that easy for you. Sorry, did you have a question? It'll come back to you. Okay. Get that synapse back. Yes. Sorry, what's the difference between CB and CR? So we can identify cells in many different ways, and sort of sadly in 2019, 2020, the way that we identify cells is so much harder for you as undergrads than it was for us. So normally we would identify it based on the morphology. So the morphology is the name. So bipolar, meaning it sends up two different projections. Or if it's a basket cell, it makes more connections. But then the protein that's expressed in that morphology can either be calretinin or it can be calbindin, right? So just like you can drive like a Honda Civic, but you can also have like a Honda Accord. So they're kind of the same, but they're a little different. Yes. So yes, so he targeted these cells. So Bruce Posner targeted these PD-containing inhibitory interneurons to control excitation broadly across the entire cortex. So remember, this is a cortical layer. So this is throughout the entire cortex. Was that clear for everyone? This is in the cortex. Frontal cortex has 6 layers. At each of these layers, we have parvalbumin-containing cells. So yes, this is how he controlled them. All right, let's see if I have one more of the, not sure, let me just check. So from today's lecture, what section do you think will be tested the most? Like you can type out your answer. Everything. Nice. Nope, not everything. Placebo. Okay. Just from today's lecture. Anxiety response in mice. Nope, not so much. Meditation, maybe, yes. Placebo for sure, but that's from the last lecture. I can guarantee you it's not everything. Is there any one specific thing from today's lecture that you think would be definitely on, outside of what I told you? Like I told you a couple of things. Anything else that you think might be on? If you're trying to get into my headspace? No? So the anxiety models, you probably need to know them for the 3, one of the 3 longer answer pages. So I'll get to that in a moment. Okay, so I think we'll just kind of leave it at that, and we'll see the last one. Okay, so the last thing I'm going to ask you to tell me a little bit about, because this is important to me to kind of know. I'm not asking you how you're going to study. I'm asking you how you're going to prepare for the final weeks that are coming up. What are you going to do to get yourself ready? Not your lecture notes ready, but like you. What are you going to do? Meditate. Nice. Okay, nice. Study. Yes, I hope so. Okay, awesome. Suffer. No, don't do that. Okay, sleep and eat. Always good. So whoever wrote that, I'm looking forward to your short answer questions, but not really. Cry. Yes, try not to. Eat snacks. Optogenetics. That would be wonderful. If you get that to work for you, please come and see me. You get an automatic 100% in the course. Sleeping well, great. Studying is good. Okay, awesome. I'm hoping for a couple of things that you're going to try to do in the upcoming weeks. And it's going to, I'm guessing that some of you, if you're taking statistics, some of you actually start your exams on Saturday, right? Saturday for some of you? And so I think for myself, it'd be really great. Oh, please don't fail. No. The one thing you should do is to take the advice that Bakiza and Rena gave you. Find time to like schedule. Find time to like rest. Enjoy yourselves. All that stuff, right? So do that. Don't fail. Brooklyn 99. Okay, good. Awesome. Okay, so I'm just going to kind of leave it at that. All right. Make plans. Okay. All right. Okay. So some of those were getting kind of interesting, but also not great. Okay. So a couple of really quick things, because we're running out of time, and I don't want to take up time from the class that needs to get in here after. You're going to have only 15 multiple choice test questions. They're going to be answer choices A through D. There will be no answer choice, either A through D, that will involve none of the above statements or all of the above. So you'll have to pick from like a statement A through D. So there's none of the above. Let me make that clear. I keep getting asked this question all the time, so let me make this really clear for you too. The final exam is not cumulative. It's everything from the last term test. So what I'm planning to do, I think, if it works for you, I will have 2 different review sessions. And the first review session will be next Friday, December the 13th. Is there a better time of day for most of you? Afternoon or morning? Afternoon. Both of these will be streamed, and both of them will be recorded. They will be posted for you shortly after the review session takes place. I will supply you with review notes for both review sessions. The second one, the Monday one, is one that if you have any last-minute questions, I will be happy to answer them for you. In addition to that, I know that some of you are interacting with the course material in all sorts of interesting ways. If I give you some sample questions in advance, will you do them? Yes. When will you do them? Next week. Okay. So my plan is, if it's okay with all of you, the Thursday before our first review session, I will post like a mock final exam that is more relevant. You can see some of the previous final exams like on the exam bank, but I want to create something that's a little bit more relevant for you to give you a better idea. Some of you, if you are willing to share your short answer questions, I will actually mark it and post it so you can see how I mark them. Would that be okay for any of you? If you're willing to, I'll anonymize it so it won't have your name on it, but if you're willing to do that. So Thursday around 12 noon, is that okay for everyone? Mock final exam, and then we will do that. So the last thing, just as a reminder, you do not have to ask me to automatically round up your final mark at the end. So as long as it's close, like if it's an 84, we will automatically round to an 85. If you're in an 89, we will round it up to like a 90. If you're at 99, we will not round you up to 100. And so outside of that, like everything else, we will kind of round you up. And I think, sorry, we're gonna do one. Oh, beach ball, nice. Okay. So I just wanted to take a couple of minutes before we take our class picture. We're all going to come up to the front in a second. Yeah, did you have a question? Sorry, are the past tests? Some of them, but to be honest, not so much. You can look at them just to get an idea, but they're not so much. Okay, one other thing. Is there anyone who's a recognized study group leader who's here? Yep, so if you are, can you come up to the front? Yeah. All right. All right, so thank you so much.
Thank you.
Thank you. All right, so I wanted to thank all of you for an excellent semester and for doing really well throughout the course. I want you to do well right through your final exams. So I do think that your class was just really brilliant. I really enjoyed coming here every Wednesday afternoon. Before we all leave, would it be okay if we took like one last class picture together? So just come up to the front. We'll kind of arrange ourselves, and it'll take like 2 minutes if we do this the right way.